Hi everyone, this is Mike, and his little brother has recently been diagnosed with autism spectrum disorder, or ASD. He's heard of the term before, but doesn't know too much about it. How common is it? How does it develop? The neuroscience behind it and its impact on individuals and their families. So let's join him on his learning journey. ASD is a complex heterogeneous neurodevelopmental disorder that usually has an onset before the age of 3. Defining ASD isn't as easy as you might think. It's not a single disorder, but a range of persistent developmental challenges in which each person is affected a little differently. Sufferers fall along a spectrum of different symptoms, challenges, and abilities that all vary in severity. Despite a diverse range of symptoms, ASD involves abnormalities in 3 core behavioural domains. These are social development issues, impaired communication skills, and restricted or repetitive behaviour patterns. Currently, there is no reliable biological test for ASD, and it has to be diagnosed by specialists based on the observation of particular behaviours. Okay, so how common is ASD? I hear Mike ask. In 2010, a global study found that an estimated 52 million people had ASD worldwide, equating to a prevalence of 1 in 132 individuals. Closer to home, the ABS estimates that in 2018, over 200,000 Australians had been diagnosed with ASD, and males were 3.5 times more likely than females to have the condition. This disorder presents substantial, lifelong challenges not only for those affected, but also their families, friends, and the wider community. Hall and et al. Estimate that a single diagnosis of ASD results in an annual national cost to Australia of between $4.5 and $7.2 billion. Whilst there are currently no treatments to cure ASD, there are several interventions that are available that may help reduce the symptoms, improve cognitive ability, and daily living skills. As such, early diagnosis and intervention is key in managing ASD in the society. Although the causes of ASD are not fully understood, there seems to be an interplay between 3 factors. Genetic, ASD is considered among the most heritable common medical conditions, with an estimated heritability reaching up to 90%. Neurobiological, which we will look at shortly, and environmental, including heavy metals and exposure to chemicals. The traits commonly associated with ASD are thought to arise due to abnormalities in how different parts of the brain form and how these brain areas are connected to each other. Through the use of post-mortem and non-invasive techniques such as MRI, abnormalities in neuroanatomy have been found in the cerebral cortex, the cerebellar region, and the limbic system in those with ASD. At birth, ASD brains appear to be normal in volume. By the ages of 2 to 4, 90% of ASD sufferers have larger than normal brain volumes. It is suggested that autistic brains tend to grow abnormally fast in early life, followed by abnormally slow or even premature arrest later on in life. The largest part of the human brain, called the cerebrum, represents the major information storage and processing region. Cerebral white and grey matter volumes in autistic toddlers have been found to be abnormally enlarged. Most overgrowth pathology occurred in the frontal cortex region. Contrastingly, the cerebellum, which is located below and behind the cerebrum, showed a decreased volume as well as a smaller ratio of cerebellum to the total brain. The most common structural abnormality is found in the hippocampus and amygdala. These 2 structures are located in the limbic system. This is a pretty impressive part of the brain that is involved in a range of behavioural and emotional responses, such as memory and modulating the learning nuances of social interactions. In fact, it has been found by damaging the amygdala of macaw monkeys in early development, autism-like symptomology can be elicited. MRI studies of the amygdala and related structures in the limbic system have shown developmental abnormalities in those with ASD. In particular, this region of the brain comprised of smaller neurons with an increased density than those in normal brains. The corpus callosum, the brain's largest and most prominent axonal pathway, is also decreased in size in those with ASD. This is speculated to lead to a disconnect between the two hemispheres, resulting in the symptoms associated with ASD. As such, it has been put forward by Hart et al. That ASD should be viewed as a developmental disconnection syndrome. In such a diverse condition, the obvious limitation to research has been the difficulties of coming to a consensus to define and properly measure ASD as a condition. Additionally, there is a substantial gap between the study of ASD from a neurodevelopmental point of view and the clinical and individual experiences of sufferers. And don't worry, Mike, the future looks good for ASD research. We may soon have the sensitive tools to diagnose ASD during the first year of life. Early diagnosis has the potential to allow for more effective teaching and educational strategies, which will increase the individual's developmental progress, reducing the lifetime costs to the individual, the family, and society.